Thank you, Holly. It's a real pleasure to be joining you at this wonderful hour of the morning in the UK. It's just gone 5 a.m. So I'm looking forward. Please accept my apologies for any sort of slight word slurring should they happen. But I've had the pleasure of being involved in the RADAAR project for the last period. This talk is actually sort of, I guess, slightly outside of what we're doing specifically in RADAAR at the moment, but it's an introduction to the potential use of genomic sequencing for AMR surveillance, something that we feel in the future is going to become more widespread and important. So I thought I'd go through a couple of introductory slides and then some examples of some projects that we're currently working in. So I'm based in between two institutes, the Big Data Institute at the University of Oxford, which is focused on developing new methods for analysing very large heterogeneous data sets, and also the Wellcome Genome Campus, home of the Wellcome Sanger Institute, which really helps develop technology. And part of our focus is on how to deliver ownership of this novel technology and data interpretation to laboratories outside of the ones that we work with. So just as some background, so one of the reasons we are interested in genomic pathogen surveillance is that in infectious disease epidemiology, more generally, one of the major questions that we try and do is, if we have a series of cases of a particular pathogen, we want to understand where they are, what's the bug, when they were individuals who were infected, who were infected, and to try and tease apart cases that are related to each other from unrelated cases elsewhere. And genomics allows us to do that unambiguously. So the process is fairly straightforward. You sample your pathogen, you run them, extract the DNA or the RNA from viruses, pass them through a genome sequencer, and then you get the genome sequence out at the end. And if you sample multiple isolates, you get multiple genomes. And then the process that we need to understand that data, bioinformatics, data interpretation follows. Now, a very simple example, if we've sequenced 3 samples, we can then compare the genomes of those samples to each other and very simply count the number of differences between the genomes. This is a very simple overview. However, these are methods that are used all the time. And once we've counted the similarities, we can then represent those graphically. It's a very simple in this kind of tree-like structure. So in this example here, the genomes from samples 2 and 3 are more similar to each other than they are from sample 1. So on this cartoon tree figure at the moment, 2 and 3 are closer together. And this is how we build up kind of relationships and visual representations of data. And then we can use that as a framework to map other pieces of information against. So for example, these are 4 categorical variables that happen to be present or absent based on, and these could be, for example, resistance to an antibiotic, but it could also be any other kind of piece of information that we might want to map to try and understand, is there something in common between the genomes that are more similar from those that aren't. So you can see here very visually, 2 and 3 genomically are more similar, and they also have these 4 things in common with each other. And then we can overlay and bring in other information, such as the geography, the location of where samples have been picked up from. So again, in this case, 2 and 3 are from a more similar location than 1. They're genomically more similar, and they have these 4 traits in common. So we're building up, a hypothesis, a risk hypothesis that there's something here that's in common between 2 and 3. And then if we overlay other information, such as time of sampling, so a very simple cartoon of a timeline here at the bottom, that sample 1 was captured and previous to sample 2 and 3, which have been sampled at the same time. So this is standard kind of questions that are asked of these data that help us build up our understanding and assessment of the risk of these samples. And this is what happens all the time. This is the basic sort of methods of genomic epidemiology. So this is a real data set. Excuse me. This is actually a data set from Europe, from Staphylococcus aureus. The red samples are methicillin-resistant Staph aureus, MRSA. This is actually from a particular epidemic lineage called EMRSA15, which is characterized previously as having arisen from the UK due to acquisition of resistance to fluoroquinolones. And we can see on the map that there are 3 geographic clusters of this red MRSA, which are all genomically more similar here. So red represents MRSA, green is MSSA, which is the susceptible background population. So this is indicative of and the emergence of this clone, this MRSA clone in the UK, and then independent introductions into both Germany and Portugal. And there are studies that have shown that this is the route that this epidemic lineage arose from. So these are the kind of tools and visualizations that we bring this information together for us to be able to interpret more easily. And these trees are used for all sorts of investigations, and you can remap those trees in different ways. So each of these tree visualizations is exactly the same data. There's no change in the relationship, but they're just rendered differently. And the more ways you can view this information, the better we can bring and overlay our hypothesis on top. So, for example, this bottom left-hand tree is essentially is quite a familiar shape. If you look into these, this is kind of what an outbreak looks like. That you have long branches that represent a background population, and then you have an emergence of something that is, short branches that is a rapid explosion. So this is that EMRSA clone, epidemic clone, that these are susceptible, widespread background population, that suddenly something comes into the population that causes the emergence and the rapid spread of a lineage. And in this case, it was, as I mentioned, acquisition to fluoroquinolone resistance. There was a specific mutation that came into the population and then caused this rapid expansion that originated from the UK and spread elsewhere. So these are kind of visual memes that you start seeing more frequently when you get into this data. But one of the important components is that context is really key. So having this background data, so ongoing surveillance of kind of the normal population, then allows you to spot the emergence of these more high risk public health focused explosions that we need to get under control. So just a couple of applications and examples of this. So one in AMR and second in COVID, because I think we're all hyper aware of the fact that genomics has been very fundamental in us identifying lineages within COVID-19. And there's a huge amount of investment and understanding that we need to upscale. And I think we need to leverage that to continue to understand and spot these kind of emergencies of risks within AMR. So work that's been ongoing in the Philippines for the last number of years. This is led by Celia Carlos at the RITM. So Celia has run a 30-year AMR surveillance program that consists of phenotypic sentinel sites, sentinel hospitals across the Philippines, who focus on WHO priority pathogens and report on an annual basis the increase or decrease in bug drug resistances for a number of bugs and a number of drugs, which are then reported upwards towards GLAS and in WHO. It's a tremendously successful initiative. Each of the priority sites store their phenotypic information within WHONET, and that is collated together to produce annual reports. So the process, to some extent, is that isolates from around the sentinel sites are sampled and phenotyped locally, and then those resistance data are sent to the central RITM lab, which are collated to produce these kind of ongoing metrics of increase or decrease in resistance. And this information is used to inform policy at the local level, at the hospital, at the national level, at the Department of Health through RITM, and then upwards towards initiatives such as GLAS. And this is the kind of figures that are produced on an annual basis, which shows you the increase or decrease to specific bug drug combinations over periods of time. Now, one of the things that genomics allows us to do is to understand, with this sort of line increase in resistance, there's no information around whether that is a single lineage. Is it a specific variant? Is it a variant that happens to be emerging or coming into the Philippines from outside, or is it spreading from north and south, etc.? So we worked with Celia to essentially transfer the ownership of the technology and the expertise so that Celia and her group would be able to look at this kind of information in an ongoing fashion using the enhanced lens that you get from genome sequencing. And what Celia, whoops, excuse me, apologies. So as a parallel process. So what happened was we worked with WHO with WHONET data to trigger sets of sequences to be undertaken based on uniqueness of resistance profiles. So this is, how do we understand the population biology of the bugs going across the country? So genome sequencing has come in here that then allows Celia and her team to understand the branches, whether there are particular strains, so operational unit or clones or lineages of these pathogens, identifying the AMR genes that are involved, and are they present in all of the strains of that particular bug, and also the vehicle that's delivering those genes. So for Enterobacteriaceae, for example, it's not as simple as a single strain is producing or is resistant and transmitting around various locations, but there's also mobile genetic elements that allow antibiotic resistance mechanisms to be inserted within a species or across species. So Celia and her team now use this to understand whether there are particular variants, let's use the parlance that we all understand around COVID, that can help inform better understanding of whether there are local outbreaks of a particular strain or lineage of a particular pathogen, and use this to inform, again, these kind of surveillance stakeholder levels. So Celia and her team have been able to use this information, represent it, to understand that they're, and have spotted local outbreaks, ongoing outbreaks of bugs such as Klebsiella pneumoniae within hospitals that haven't been spotted using the standard phenotypic surveillance. So this has allowed then infection control practices to be put into place, and there are some increased descriptions of this work in the publication here. But it's really starting to help transform the ability to spot and stop outbreaks much more rapidly. And this work that Celia has driven is being expanded to focus on technical support for the implementation of the technology, the genome sequencing, but also the data interpretation and those methods that I've just very briefly touched on. This is a project that's been ongoing for three years with groups in India, Nigeria, and Colombia. So each institution represents a broader network of sites that are collecting phenotypic information and are now getting much more resolute information using genomics, along with scalable technical support packages for setting up genomics within clinical microbiology laboratories, external and internal quality assurance exercises, training, and shared learning across each of the 4 sites who are taking this technology on. So as an example, in Nigeria, RUCA has really led the development of the Nigerian Action Plan, but the set of Sentinel sites are much less mature than those in the Philippines. But Iruka's team are learning an awful lot about the 30 years of phenotypic surveillance that has been ongoing in the Philippines, so that they can drive down the amount of time it takes them to get to the stage that Celia and her team in the Philippines are at the moment. So the really important part of this is the shared horizontal learning across sites. But of course, very, very simple examples, and there's a lot more detail to this. There are major bottlenecks, and those bottlenecks tend to fall back around data and data tools. How do you link genomics to epidemiology and other clinical information in the local sites? And then this rapid interpretation on top of challenges around political buy-in from institutes. It's a nice technology. We can show simple examples, but to get that into common practice, we need to have buy-in at an institution, but also a governmental level. And issues with supply chains for reagents and costs of undertaking these methods. So just to switch very briefly to COVID-19. So similar issues. As the pandemic started, we require the ability to understand whether it's a single homogeneous population or whether there are variants that are emerging that could cause concern. And we're all aware of the variants that are causing concern across the globe at the moment. So in the UK, we were lucky enough to fire off a project that aimed to roll out sequencing across the country as rapidly as possible. This was a project called COG UK. And the idea was to have decentralized sequencing sites across the country that would sequence as many of the positive samples as possible, all of that data centralized and analyzed, and then the trees and the interpretation to be sent out to each of the sites so that additional analysis can be undertaken. So this was a very vast project involving many, many institutes that took on the sequencing themselves locally, but to bring that data together nationally to be able to understand what's going on. Open data sharing. So all of the genome sequencing was put into public archives and then data linked to as much of the Resolute epi and patient information as possible, along with information around, for example, mobile data or movement patterns to understand whether there were certain lineages or variants that were transmitting due to population movements, and then contact tracing, etc. And another key thing that's come out of this is the rapid identification of these lineages, these variants that are emerging all the time. So big trees are produced. So currently, I think we have globally well over 1 million genome sequences. And once you start to do that method that I showed you about comparing them all together, your trees get really bloody big and really difficult to interpret. They become essentially useless to be able to look at a single tree. It's not a way of looking at data of this size. So breaking big trees down into smaller branches that are manageable units is essentially how you define these lineages. So Andrew Rambow and his team in Edinburgh came up with this naming scheme called Pangolin, which essentially assigns IDs to specific branches. So in this case, this is the B117, which we're all aware of, the alpha variant, as it's termed now, due to WHO. So this just allows big, massive trees of data sets to be described in ways that are available for public health labs to talk about the same thing across the world. And then data can be presented in these visual fashions. So this is samples from across the UK, and this is over time the emergence of lineages. So beginning of the pandemic all the way through, you can see that there's a very big red splurge here that is then supplanted by this very big green section here. So this green section is the alpha lineage that became rapidly the dominant lineage in the UK. Again, bottlenecks here are data and data tools, linking genomics to epi, rapid interpretation, etc. However, there are similar data concepts going on across SARS-CoV-2, AMR, with genomics, that the trees, geographic information, temporal information, and epidemiological variables need to come together rapidly for us to understand and ask the epidemiological questions that we want to identify. Is there emergent risky lineages coming? Where are they, and how can we track them and stop them spreading? So just to say that there are common data processes when looking and using genomics for COVID that we apply within AMR that can be applied to any sort of pathogen paradigm. For example, if you take a genome sequence, be it RNA or DNA, identifying what the species is, and then if you understand the species, then you can look for risk markers such as specific mutations that define a lineage for COVID, SARS-CoV-2, or molecular typing methods such as MLST, CG-MLST. Within Salmonella typhi, there are specific typing methods that can be used to identify from DNA sequences whether your Salmonella typhi is of a particular high-risk lineage or not. And then additional risk markers such as specific mutations in SARS-CoV-2 genome that may well indicate that there's potential vaccine escape, for example, or AMR determinants. So it's still, the basic premise is identifying specific signatures in the DNA that can give us additional information that we can assess the risk of that particular sequence. And this is conceptually the same across viruses, bacteria, fungi. So it's very important that this technology is pivotable across pathogen paradigms. And of course, we focus on the critical priorities. We have a route to delivering sequencing within the current global and national action plan routes. And I thoroughly recommend reading the GLASS document that goes into some of the problematic aspects, such as supply chains, cost, actually what you get from the genome sequences that can be used to gain buy-in. And I think we're at a pivotal moment right now where there's a lot of focus and enhanced investment into using this kind of technology for COVID-19, which we need to adapt to the legacies that we can return and pivot towards AMR. I think it's a very powerful powerful tool. And I'll finish there. Thank you very much, Holly and Marianne.